besides what we see in the face. So there's generalized hirsutism, means there's a lot of body hair. They're usually small babies, even when they're in the womb. But even after they're born, they usually don't grow quite as much as the other kids. They're hypotonic, means their muscles are not quite as strong. And they have feeding problems, especially a common thing in congenital diaphragmatic patients is the reflux. Mainly because there's probably a change, an abnormality in the muscles that close the esophagus. And there is a developmental delay. So I mentioned the limb before. The limb is very interesting, and it's basically the reason why I chose to talk about Cornelia de Lange. So these are the limb findings in these babies. There's micromelia, so it means relatively smallness of the hands or feet, which is almost universal. It means almost every baby that has this diagnosis has it. Sometimes this micromelia is really apparent. You'll see a picture later. You can't miss it. Sometimes the smallness is relative, and this is why taking measures can help get to the right diagnosis. There may also be more severe abnormalities like oligodactyly, which means missing digits, and less common limb findings. And this is striking ones. A single palmar flexion crease, so there's only one line in the hand. A fifth finger clinodactyly, which is a change in the fifth finger. Proximally placed thumbs, so thumbs are usually a little bit lower than they would be. And partially syndactyly, so there might be a little bit of skin between the fingers. This is the most striking aspect that this syndrome can get to. So we've seen an autosomal dominant syndrome, and we've seen the limb defects in that syndrome. The reason why we're concerned with the limbs is from an embryological point of view, because we realized studying embryology that the limb and the diaphragm form about at the same time, a little bit earlier the limbs than the diaphragm does, and they share a little bit of common mechanisms in their outgrowth from the trunk. So of course the limb butts out of the trunk, grows outside. The primordial diaphragm, which is called PPF, grows off the body trunk but inside. And some of the mechanisms that drive this growth seem to be analogous. There's not much that's known about how the PPF grows, but Meaghan is doing a lot of embryology research in that. But there's a lot that's known about the outgrowth of the limb. So if we can tie them together and draw analogies, it really can speed up the research and know what to look at. This is the other syndrome that I'm going to talk about today, which is called Donnai-Barrow. Its story is very, very long. It was first described in 1972 by these two very good geneticists, Dr. Holmes and Dr. Shippens. They described these two siblings with a characteristic array of birth defects, including CDH. In their paper, they suggested that this is a new syndrome of multiple anomalies, the model of inheritance of which is possibly autosomal recessive. This is a completely different mechanism than the autosomal dominant we saw before. In this case, each of the two copies of the same gene have to be mutated, to carry the mutation in order for the phenotype to be expressed, right? So in this case, usually the parents don't display the phenotype, don't have the disorder. After Dr. Holmes and Dr. Shippens made their description, more than 24 similar patients have been reported. The most notable description was by Dr. Diane Donnai in the UK. For this reason, now we know the syndrome as Donnai-Barrow, Barrow being her collaborator. And this condition has been variably reported as DBS and 4 syndrome. Now 4 is not exactly the same as DBS, but now we know they have the same genetic lesion and their similarities are a lot. This is what Donnai-Barrow patients have. They have a diaphragmatic hernia. They have an abdominal wall defect that we call exomphalos. They have a genesis of the corpus callosum. I have a picture later that shows what that is. They have severe sensorineural deafness, severe myopia, occasionally retinal detachment, and proteinuria, which means that if you look at the urine, you find a lot of proteins are being spilled. So they're wasting a lot of proteins in the urine, because their kidney simply cannot reuptake those and put them back in the blood where they belong, which is what the kidneys normally do. Also, the Naibero patients have a very characteristic facial feature, including hypertelorism, which is the increased distance between the eyes. Also, the palpebral fishes have a particular shape. The nose is short and bulbous, and the ears are slightly posteriorly rotated, which are placed like this. And this is what that looks like. You see, bulbous nose and the hypertelorism you see. And this is the brain change that I mentioned before. So the agenesis of the corpus callosum. There is this part of the brain that you can see in this drawing. This white matter in the center of the brain. This is a brain that's been cut midway and reproducted this way. This is called the corpus callosum. And it's basically a bundle of nerves that connects the right half of the brain and the left half of the brain, back and forth. And in these patients, it's either entirely missing or severely reduced, so that it is hypoplastic. Like you see in this MRI, you see the back part is missing altogether. And even the front part is severely reduced in its size. So DBS definitely does run in families, may run in families. There are 17 individuals in 15 families that have been published. And in 2 families, there are more than 3 affected members. And one of the issues with families that have the Naibero syndrome is consanguinity. This means that the parents are related with one another. This is the reason why we see a lot of Naibero syndrome in consanguineous parents is because the mutation is so rare that the odds of 2 unrelated person marrying and giving both the mutated genes to their offspring is just very, very strange. We do find that, we do see that, but the majority of those families are consanguineous. And a large consanguineous family is also a very important tool in research because it allows you to use a genetic kind of analysis that's very powerful. It's called homozygosity mapping. So if we look at this picture, this is what homozygosity mapping is. So let's assume that the gene with carrying the mutation is in this region of the chromosome. This is dead. You can see the picture. Put the picture that you can see. It's in this region. Mom is also carrying a region with the mutation in it. And they have 3 kids. They have Arthur that has both the regions and will have Naibero syndrome. Casper has both regions and will have Naibero syndrome. And Josephine only have 1 region because maybe the mother passed on this chromosome and the father passed on the green one, or vice versa. So what we do is we put landmarks throughout the chromosomes. We look at letters throughout the chromosomes, and we compare all the chromosomes and say, is there a region that is the same? Because this region has to be the same here and here, right? Here and here. Because they carry on the same, being consanguineous, they carry down the same stretch of DNA. And within that region that looks the same, there might be a lot of genes, and you can sequence all of those genes looking for the actual mutation. And this is what has been done by Sibel Kantarch in our lab.